Have you ever really stopped to think about what a virus is? Not just as some germ that makes you sick, but as a machine, a microscopic invader with one single goal. Today, we're going to tear down a plant virus, piece by piece, to see how it's built from the ground up. All right, let's get into it. So, let's start with the big question. How do you build a microscopic hijacker, something so tiny yet so ridiculously good at taking over a living cell? It's kind of wild when you think about it. This isn't just random biology. You know, it's like a masterclass in minimalist design.
And you know what? The answer is shockingly simple. It really just boils down to 2, maybe 3 parts, built from the inside out. You've got the blueprint, the shell, and then sometimes a stolen cloak. And yeah, we're going to break down every single one.
Okay. First up, let's start at the very core, the absolute heart of the whole operation, the genetic blueprint. This is the instruction manual, the software that tells the virus exactly what to do to hijack a cell and make more of itself.
So right away, we hit the first big design choice. Is the blueprint going to be written in DNA or RNA? And it's always one or the other, never both. Now for us humans, DNA is like the big master library, and RNA is the little note you send out to get stuff done. But for a ton of viruses, especially plant viruses, RNA is the master copy. It's like they've got a faster, leaner operating system.
And then there's another wrinkle. Is that code single-stranded? Think of it like a single piece of tape. Or is it double-stranded? You know, like that classic DNA helix we all know. This choice makes a huge difference in how the virus has to hack into the cell systems to get its instructions read.
Okay, but with RNA viruses, it gets even cooler. If it's a positive-sense RNA virus, that means its instructions are basically ready to go. The cell can read it immediately and start making virus parts. But if it's negative-sense RNA, that's like an encrypted file. The virus has to bring its own password, its own little decoder key, to unlock the instructions first. It's a really slick strategy.
And the whole point of all this is efficiency. Just absolute, ruthless efficiency. Viral genomes are tiny, like ridiculously small. Some have just enough code for a few proteins. Get this, the smallest one we know of codes for just one single protein. That's it. Mind-blowing, right?
All right, so we've got the blueprint, the software. But you can't just leave that lying around, right? You got to protect it. So let's move one layer out to the shell. And you can really think of this as a custom-built, self-assembling suit of armor.
This shell is called the capsid, and it's made up of tons and tons of identical protein subunits. But here's the brilliant part. These little protein pieces are designed to just snap together, all on their own. They automatically form this perfect container around the genetic code, like a bunch of magnetic Legos. No assembly line required.
And these self-building shells come in a few basic shapes. You've got these cool rod-shaped ones that look like a tiny spiral staircase. Then there are the isometric ones, which are basically these super efficient 20-sided shapes that look a lot like a soccer ball. And then you get the really weird ones, the complex ones like Gemini viruses, which are literally 2 of those soccer balls fused together. It's all about what shape works best for the job.
But this suit of armor is way more than just protection. The capsid is like a Swiss Army knife. It's the handle that an insect, like an aphid, uses to carry the virus. It's the key that lets the virus move between plant cells. It even knows the exact right moment to pop open and release the blueprint. Oh, and it can also fight back, shutting down the plant's own defenses. This thing is a seriously sophisticated piece of tech.
All right, on to our final layer. Now this one's an optional extra. Not all viruses have it. This is the cloak. And the craziest part? The virus doesn't even build it. It steals it.
So an enveloped virus is a virus that wraps its capsid in a fatty membrane. And where does it get this membrane? It literally steals it from the host cell as it escapes. It's a perfect disguise, a piece of the host, but dotted with the virus's own special proteins.
So how does this heist go down? It's a pretty cool process. First, the core of the virus gets assembled inside the cell. Then it drifts over to one of the cell's own membranes. From there, it just pushes its way through, almost like blowing a bubble. And as it pushes out, it wraps itself in that membrane. And voila, it emerges on the other side, totally cloaked and ready to go.
But why bother with all that? Well, that stolen cloak is basically its passport. Those little viral proteins stuck in the cloak, you know, the spikes, they're like a key. They're what allow an insect vector to grab onto the virus and give it a ride to the next host. Without that cloak, it's grounded.
So let's pull back for a second and look at the big picture.
What's amazing here is how you get all this incredible diversity from just a few simple mix-and-match parts. It's really a masterclass in biological architecture.
You can see how it all builds up in layers. At the most basic level, all you really need is the blueprint. But of course, most viruses add that protective shell, and then the most kitted out ones add that final stolen cloak. Each new layer gives it new powers, new ways to survive and get around.
And it all comes down to this one simple idea, which is just perfect. From just a simple code, a protein shell, and a stolen membrane, you get this absolutely staggering variety of biological machines. And every single one is perfectly tuned for its specific job. It is the ultimate example of doing more with less.
So there you have it. The virus is a masterpiece of minimalism. It's built from the simplest parts, but it's endlessly adaptable and just relentlessly efficient. It really makes you wonder, doesn't it? What new designs, what clever new tricks are these tiny little architects going to come up with next?